everyone. So in this video we are going to study about hybridoma technology, which is a very important technology used in today's world in the field of biotechnology, life sciences and other medical processes. So the name suggests hybridoma, which means in this process we are going to produce hybrid of something. What is that something? The cells. It is a technique which is used to produce large quantity of specific monoclonal antibodies. Specific means which have a specific function, one type of specific monoclonal antibodies which are produced in large. It combines the property of B lymphocytes, where these B lymphocytes come from B cells, which provide antibodies with the ability of cancer cells to divide indefinitely. So what does this process do? It combines the property of B cells, which provides antibodies with the ability of cancer cells. What does cancer cell do? Cancer cells have the ability to divide continuously, uncontrollably and indefinitely. So when we combine the property of B lymphocyte with antibodies, they become indefinitely divided. They continuously divide and produce large quantity of monoclonal antibodies as the cancer cells. So the resulting fused cell is called hybridoma. So when an antibody is combined with what? B lymphocytes. The property of B lymphocytes, it becomes very able to produce large number of cells, just like cancer cells. I hope you got this point. So when we combine these two, the antibodies with the property of B lymphocytes, a fused cell is formed, which is called as hybridoma. And the process used to produce this hybridoma is called as hybridoma technology. So let's see the history of this hybridoma technology. I have mentioned only the name and the year. You can read it more. Like first time it was developed by Georges Scholar and Cesar Milstein in the year 1975. So these two were the developer of this hybridoma technology. The hybridoma technology is a long process which involves various steps, as you can see here. The first step is immunization, the second one is cell fusion, third is selection, fourth is screening, fifth is cloning and sixth is antibody production. So all these steps are done why? To produce monoclonal antibodies which are produced at the last step. First, the immunization process is done, which leads to cell fusion, and when the cell fusion is done, we select the specific type of cell. As I told you here that specific monoclonal antibodies needs to be produced, so we select specific type of cell from here. We do the screening of that cells. Once we find that the specific cell are good enough to carry out the process, we do the cloning of the cell and produce large quantity of antibodies, and then the antibody production is done. So these 6 steps are involved in the hybridoma technology. What is the use or application? So the first use is in the diagnostic test. We see diagnostic tests are performed in the labs where hybridoma technology is used. For example, you have only one type of cell. You got only one type of cell from a body and you cannot further isolate those cells. So you can use this hybridoma technology to make many cells from one type of cell and do the specific test or function. Next is therapeutics. It is also used in making therapeutic drugs, carrying out therapeutic process to detect a disease. The third is research tool. It is used in various research where we have less number of cell and then for target therapies for disease such as cancer, autoimmune disease, infectious disease, etc. So this was all about hybridoma technology. Let me know if you need to study all this in detail. Thank you.